Welcome to the National PKU Alliance funded research webinar. By way of introduction, I'd like to talk just a moment about the National PKU Alliance. The mission is to improve the lives of individuals with PKU and pursue a cure. The National PKU Alliance works towards these goals by assembling a world-class scientific advisory board who helps the NPKUA identify new cutting-edge directions of basic and medical research, funding new and promising research at major universities around the world through a competitive application process based on scientific merit, funding postdoctoral fellows who bring fresh insights into PKU research, funding the development of new technologies, such as the home fee monitor, to help PKUers better manage their care and accelerate medical research. To date, through the generosity of donors, the NPKUA has funded a little over $4 million in PKU research and postdoctoral fellows, which has led to a deeper understanding of PKU and the development of potential new treatments. Next slide, please. Today's presenters are a sampling of the research that the National PKU Alliance has sponsored. First, we'll hear from Shawn Christ, PhD at the University of Missouri, talking about advancing our understanding of PKU in the brain. Then Roberto Gramignoli, PhD at the Karolinska Institute in Stockholm, Sweden, who will be talking about new cell-based therapies. Carrie Harding, MD at the Oregon Health and Science University, who will be talking about gene editing approaches to PKU. Erik Koppes, PhD at the University of Pittsburgh, talking about genome-edited swine and cell culture models of PKU. And then finally, Desirée White, PhD, Washington University in St. Louis, who will be talking to us about clinical drug trial endpoints. We will have a question and answer session at the end of all the presentations. If you'd like, you can enter your questions through the chat box that you'll find an icon at the bottom of your screen called chat. And when we finish the final presentation, we will start working our way through the questions. So we're excited to bring these researchers to the attention of the NPKUA community. And I'd like to start off with Shawn Christ. Shawn? Hey, Shawn, you might be on mute.
Unmute now. All right.
Okay.
I lost control of my computer for a moment. So again, I'm honored to be here today to share an update with everyone about the research that we've been doing that is graciously funded by NPKUA. Research, oops, let's see. We're hitting our bugs already, aren't we? Back up. All right, there we go. Research in our laboratory focuses, what we focus on is the brain. And the brain is a very complex organ. But one of the basic distinctions we can make in terms of the brain is between what we call gray matter and what we call white matter. So we can think of gray matter as that tissue where the data is processed. And in contrast, the white matter represents these long distance connections that allow for the communication of information between disparate brain regions. And of course, the integrity of the gray matter and the white matter can go together and contribute to cognitive performance and how we do. In terms of PKU, what we're interested in is how PKU, or kind of more focused at the excess phenylalanine associated with PKU at times, can affect gray matter and white matter and therefore affect cognitive ability. So within that context, what the research we're doing right now is trying to understand the dynamic between these different factors. So, how does the integrity of the gray matter contribute, or or combine with the integrity of the white matter to predict cognitive performance. And so the study that we're doing right now is we're collecting a large sample, data from a large sample of adults with and without PKU, and getting an extensive cognitive and neuroimaging brain battery. I'll just kind of go through this. So far, we're almost done with data collection. The COVID situation kind of put a halt on our research, as well as I imagine many people. But we've got 37 adults with PKU that have participated, 33 without PKU. You can see that they're fairly well matched in terms of age and other things. Our goal is to get to 40 individuals with PKU, so we're very close on this. The battery, as I mentioned earlier, we really want to get a really full, comprehensive picture of cognitive ability, strengths, and weaknesses in our sample. And so we're looking at a number of different abilities, including things like executive function, motor control, even emotional regulations. So propensity possibly for anxiety or depression, and so on, processing speed, attention, and other things. In terms of our neuroimaging battery, that's also quite extensive. One of the things we're doing is, in order to look at the integrity of the gray matter, we're using what are called high resolution T1 imaging. So this is very high resolution, so down to 1 millimeter. As you can see from the picture on the screen, this is much as what you might imagine, or we've all seen it in terms of an MRI, but again, just very high resolution. In terms of the white matter, we take a different approach, and we use a different methodology called diffusion tensor imaging. And for brevity, I'm not going to go into the nuances of that, but I did want to highlight one advancement that we're using right now in this study that I think is very interesting and cool. And it's called super resolution imaging. And so what we're doing is, this is based off of work that's been done in the visual field with two-dimensional images. And so what we know is, if you have a number of two-dimensional images that may be low resolution, so in other words fuzzy, but they're all the same image, you can actually combine those to create a high resolution clear image. And so much the same way, we're doing that in 3 dimensions with the brain. So we'll acquire this data, in this case in 2 millimeter slices, and get a multiple of those, and then we combine those together, and we're able to get a super resolution, so 1 millimeter or below, or above, I guess you'd say, resolution image. And so this is kind of an advancement in doing this in 3 dimensions with imaging data versus 2-dimensional camera data. So just to kind of give you some preliminary results, very preliminary, in looking at the cognitive battery, what we're seeing is group-related differences on a number of measures. So subtle differences on executive function, processing speed, motor control, sustained attention. These are things that we know from the literature. Individuals with PKU that have higher Phe levels can be susceptible to having difficulties with. So not overly surprising in and of itself. I did want to just, instead of showing you all the data, again, we're kind of limited on time, to just highlight the sustained attention component. So with the sustained attention, what we use is a task called a continuous performance task. And it's boring, it's simple, it's meant to be boring. So it takes about 20 minutes, and you're simply pushing a button on a computer whenever a certain letter appears on the screen. And so you can imagine at certain times you're pushing the button a lot. At other times, you may be sitting there for a while waiting for the target. So it can get at impulsivity, but it can also get at sustained attention. And we look at the results here just to kind of orient you. On the x-axis on the bottom, you have these different measures, and different measures that come out of this test. The 2 we want to pay attention to are the second and third ones, the omissions and commissions. And on your y-axis, you have standard score, higher is better. So omissions are kind of misses, like you were supposed to push the button but didn't. So that's kind of an indicative of difficulties with sustained attention. So we'd say it's inattention. The commissions on the other side are more impulsivity. So you're pushing the button, the target comes up, you shouldn't push it, and you accidentally do. And so what's interesting here is where we saw the group differences really was in the omissions, so the inattention. So this is kind of consistent with the idea that, again, individuals with PKU, if they're susceptible to attentional difficulties, they tend to be on the sustained attention side or inattention side more so than the hyperactive side. Doesn't mean that you don't have individual variability, definitely, but just again, at a group level, this is what we're seeing. Now, from the brain imaging side, we're still processing much of this data, but I did want to share this. We actually just finished analyzing this the other day. Thank goodness for one of our team members, Alex Brown, has been working his butt off on this. So what we have here is we're looking at the gray matter. And this is for our group. Every place that you see that's blue is showing a significant decrease in gray matter thickness. So what's most interesting about this is that we had a paper come out 4 or 5 years ago, which was a slightly smaller sample, I think about 20 individuals with and without PKU. And we also saw this kind of cortical thinning. And the big part was it tended to be in the posterior part of the brain, so the back of the brain. And what catches me about this is that just my first initial look at this data, because again, we've had it for a total of 1 day, is that this is also towards the posterior brain. So it's kind of nice because it's replicating and generalizing what we found in our other study. The other thing that's kind of interesting here, and I have a little circle that comes up here. Again, in that older study, everything was in the back of the brain. In this one, we're seeing some slight differences also towards the front of the brain. So in this case, this region that's kind of circled is in what we call the inferior frontal gyrus. And so this is part of the prefrontal cortex that's important in things like working memory and inhibition. So again, we're going to finish analyzing this data, go through it, but very interesting. So looking forward, so we're almost there, as I said, with the study. We have to collect final data on a handful of participants. We've got 3 more people that we want to run. We also need to finish up processing on that white matter DTI data. That super resolution methodology I talked about is actually quite computer processing heavy, and so we're doing our best. Also, we have to gather remaining fee records. So right now it's a tough time for everybody, including clinics, and so really we're trying to work with clinics to get these historical fee records so we can say, like, hey, if we see a difference in gray matter or white matter, is that related to fee levels now, or when the person was 10, or when they were 3, or when they were 1? And so it's an important question to kind of ask and answer. Again, our ultimate goal is to bring all these pieces of data together to say, like, okay, that's great. We see differences in gray matter, but what does it mean? Does it have any relationship to the differences in cognition that we're seeing? So we're seeing difference in gray matter. Does that relate to the differences in white matter we may see? Off for a minute. Oh.
Okay.
Sorry, I'm back on. Was I out there for a little bit, Lex?
You were froze for just a moment. Did Michelle? Can we go back to the last slide real quick?
Let's see.
One more. There we go.
Oh yeah, this is actually the most important one. This is the one where I say thank you to our team. Just to give you guys, I know it's been an impact. This spring, before the COVID hit, we had 15 people working hard on this data, and then COVID hit, and of course the university shut down, and so it's really been a mixture of Alex, Haley, Emily, and myself, and a few others just trying to put this together. But again, thank you. Can't do it without our team.
Okay, thank you, Shawn. That's very exciting, and I'm hopeful that in addition to bringing insights into you know, how the fee interacts with the brain, that your observations may also contribute more broadly to the understanding of brain structure and neurocognitive function. I'd like to remind the audience that if you have any questions for Shawn, we'll be taking questions at the end, and you may submit your questions through the chat function of the webinar today. Next up is Roberto Gramignoli, who is at the Karolinska Institute in Sweden. Roberto is doing some very exciting work developing cell therapies for the treatment of PKU, but also these may be broadly applicable to a number of other diseases too. Roberto, welcome to the seminar, and we're excited to hear about your work.
Thank you. I think you can hear me fine. So thanks everybody. Good morning, everybody, and welcome to the other side of the ocean. So I'm taking a few minutes of your time this morning to illustrate, to go through all the results we have achieved thanks to your generous support during the past year. So as you might know, we are actually working in cell-based therapy. So what does it mean? We inject the cells into the liver in order to correct different liver disorder. We bridging patient to the organ transplant, but we can also rescue patients with acute failure with different disease, including PKU, different metabolic disorder. As you saw, even before and now, we injecting the cell through the umbilical vein, delivering them into the liver. And once they are inside the liver, they are capable and functional in correcting the disorder. Which metabolic disorder we have been transplanted in patients so far? In our group and also different group around the world, you can see there is quite a wide list of different disorders. And I'm happy to say there are also 2 phenylketonuria treated with transplantation of liver cells. Anyhow, transplanting liver cell, despite how successful can be, and how great can be in sort of way, is very difficult to achieve because we have very limited amount of cell we can transplant. That's why during the past year, we have been investigate every kind of cell we have our hands on. That we are moving from mature liver cell, which is expressing the enzyme to correct the PKU, but we also investigated different progenitor and stem cell in general, including embryonic or iPS cell, in order to evaluate the potential treatment of different disorders, including PKU. As you can see, the first one, for example, was the hepatocytes. Thanks to the PKU support in 2010, we were able to translate to the clinic. But the most powerful and interesting cell we identify are actually cells not isolating from the liver. Surprising are the cells we can isolate from placenta. Placenta is a very intriguing organ. I start learning more and more during my career because this is an organ which usually we don't pay much of attention, because once the baby is delivered, nobody cares about the placenta. What we and Dr. Strom actually together we identify is that this thin layer surrounding the baby, it's called amniotic or amnion membrane, is a tiny little bit of tissue. As you can see, it's composed by very large amount of extracellular matrix, which are not viable cells, but are cells, viable cells on the top facing the baby, and these are called epithelial cells, and few other cells called mesenchymal stroma cells. The MSC cell has been used in different treatments so far, but nobody has ever identified or been able to isolate this cell and investigate their potential. We developed a technique thanks to which we can isolate this cell and this cell only. You see the difference before and after the treatment. So we collect this cell and we use this cell to treat different disorders. And we propose also PKU. Why we like so much this cell? This actually cell, first of all, of course, it's quite easy to get. The placenta baby are delivered every day. But these cells are very peculiar because they have a lot of interesting properties. First of all, they're safe. They've been transplanted in human for decades, and they're never form any tumor, never have any problem. They have this kind of plasticity, so multipotency. What does it mean? They're capable to differentiate, to mature into all the cells in our human body to different extent, of course. This is under investigation by our group, other group as well. They have immunomodulatory properties, and about this I'm going to talk a little bit more later. They are anti-inflammatory, so they reverse inflammation where they're going. They have some antibacterial and some antiviral also property, which are under investigation. And probably more important for us is the pro-regenerative effect. We like so much this cell because once we injected this human cell into a mouse liver, we tested, of course, in animal first, what we find out that this cell, the brown one, are the human cell from placenta. All the other cells around are liver cell from the mouse. We didn't do anything to this cell except for putting exactly there in the liver. And what's happened? Then the liver, the surrounding cell in the liver of the mouse was capable to induce our cell to mature. And our cell became fully mature, exactly resembling the liver cell, including this tiny little bit of enzyme, PAH, which all of you are probably familiar with. So this cell becoming a real function hepatocyte, capable to reverse different metabolic disorder, but are really capable to reverse the metabolic disorder. How we can prove that? Of course, before testing in human, we want to test in animal, and that's what we did. Since we were successfully treated patient with human hepatocyte, with liver cell, we first compare injecting to the PKU animal some mouse hepatocytes. And that's the level of phenylalanine correction when you inject hepatocyte. So the same cell we injected in 2010. into the serum and in the brain, because the second bar on the right is the brain. So we measure also phenylalanine in the brain. So we have a 60% reduction in the brain and 25 in the serum. If we inject human cell from the placenta, this AE cell, what we call it, we got a more sustained, higher reduction of phenylalanine level in the blood. So the reduction of phenylalanine level was 60%, quite close to normal, not completely normalized, but quite close. And in the brain, we get even a better reduction of phenylalanine level, so 85% reduction. Still in the animal, we measure additional molecule, chemokines, and amino acid. So we measure, for example, the monoamine, because we are interested in the monoamine pathway for different neurological disorder, and the problem is correlated with the PKU, as you can find in yellow our cell. In several cases, the level of correction is very similar or totally normalized compared to the healthy person and not the sick animal, the black one. In other cases, we are not completely achieved the fully restoring. So we are still finding if we need more cell or not. So other amino acid, of course, we did several different control, but the important one right now is the one in the red bar. So our human cell injected in these mice. And you can see most of the level are strongly improved, if not completely normalized. These are very interesting cells, as I mentioned, because we are not the only one now interested in using this cell, because so far different labs around the world have been testing this cell in different organs. Of course, we are mainly interested in the liver, but we find out that actually this cell can have a beneficial effect in reversing a lot of disorder in different organs, including lungs. Nowadays, the most studied one probably because of, unfortunately, because of COVID, but kidney, uterus, pancreas, several organs can actually benefit from the therapy using this cell. But I mentioned you that these cells are immunomodulatory. What does it mean? That so far we transplant a human cell into a mouse with a complete normal immune system, which will be honestly recognize the human cell immediately within seconds, and we never find out any problem. So we started to investigate how is it possible that this cell escape the recognition and the rejection they usually characterize when we do the transplantation of an organ or other cells. So we find out that our cell express this molecule called HLA-1A. These are molecules that actually are expressed in every cell on your body. So our cell expressing as well. And when you have this kind, it's like an identity card. So what does happen? When you transplant cell, which doesn't belong to your body, is usually recognized by your immune system, by the police guy in the immune system, and usually recruiting more and more police. And that's not ending good because you are toss them out. They're usually rejected. Our cell usually we identify they have several mechanisms, particularly this HLA-1B, this little letter make a lot of difference because this molecule is actually protecting our cell. HLA-G is a molecule that usually everybody can measure in our cell and other. These are very interesting molecules because when the police guy, when the immune system is finding our cell, there's no effect. So they recognize as a protected one, not risky for the body. More important now, we went on investigating, we find out that we have additional mechanisms which this cell can actually enhance and drive regeneration in different tissue, including the liver. What does it mean? That when the cell, the immune cell, are getting in contact or in proximity of our cell showing something, so they express some molecule, or secreting as well some molecule, which educating the system to become a little bit more pro-regeneration. So these cells are not fighting against our cell, but actually educated to regenerate, to heal the organ which was received insult. Of course, a little bit, that was a nice diagram, but let me show you what does it mean. So we isolate the cell, we expose to the different immune cell, the T cell, B cell, natural killer cell, macrophages. These are the cells that most of the time are involved in your immune system to recognize tissue or cell and rejecting them or working against them. And when our cells are getting in contact with this immune cell from a donor, so they are different people, we find out that the macrophages switch into a pro-regeneration phenotype. That means they are working not against our cell, but against the disease. The NK decrease proliferation, so they don't create any problem. The T and the B cell switch into a phenotype, so an expression of a peculiar kind of working expression, which is called regulatory. So Treg and Breg. Regulatory is good for us because that means it's pro-regeneration most of the time. Okay, I'm reaching the conclusion because probably the time is quite over, but I want to show you just a couple of things about what we are actually working nowadays in the lab, despite this virus slowing down several activity. So we proved that these cells are safe, not tumorigenic. We proved that we can isolate this cell thanks to the PKU support, to the NPKUA support. We can isolate this cell in GMP conditions, so means in clinical grade, and produce cell ready to be transplanted. We validate how to inject this cell for liver or for other organ, but especially in liver. We proved that we can correct different disorders, including PKU in animal. We got permission to transplant the first-time patient in Karolinska. And now we have strong evidence that we can inject, and why we can inject this cell without immunosuppression, which, by the way, nobody wants, but we try to achieve on that. What we're going now, what we actually, during the past month, we saw was the problem related to the cryogenic preservation. What does it mean? That we usually isolate the cell, but we want to freeze the cell. So usually we have different placenta and we freeze in bag, ready to transplant in patient if it's required. But in order to keep it for the time we get the patient ready, we need to freeze this cell. And banking and storing in this huge cryotank, as you can see, ready to be offered to the patient when it's needed. And of course, we are quite good in that because we do not only freeze the cell, but we guarantee that the cells are the best quality we can produce because we tested for function and characteristic of this cell. Most of the time, guys, and everybody asking us how many cells we need, of course, the amount of cells we need to inject in the patient is related to the age and to the type of disorder we need to treat, because it's different disorder require different amount of cell. But definitely if you are a baby, newborn baby require very few cells compared to an adult, because the body mass is quite big. So you can realize that we need plenty of this cell potential for an adult. How can we solve this problem with so many cells we need? We can use different placenta, absolutely, that's a strategy because placenta are readily available, so it's no problem. Or in alternative, that's what we are doing. We try to expand the cell from one placenta to generate bags and bags, multiple bags from the same placenta in order to inject 1 or 2 donor max to a single patient. And we are quite, we are struggling to move into the GMP condition, so we try to translate into the clinic as soon as possible. So even for me, this is the most important slide. So the very last one, as you can see, this is a project was starting in the early 2010 in Pittsburgh, so in your side. And then when both me and Dr. Strong, we relocated in Karolinska, we keep working on this thanks to the collaborative and your generous support. So I would like to thank you, Gord, and I'll take any question after all this session. Thank you.
Thank you, Roberto, for a very interesting discussion on a new approach for the treatment of PKU, and in fact, again, may be more broadly applicable to lots of diseases. But thank you again. And if you have any questions for Dr. Gramignoli, please send them through the chat function. Next up is Dr. Cary Harding from the Oregon Health Science University, and he's going to be talking to us today about gene editing approaches to PKU. Cary?
Good morning. Thanks, Lex. Just waiting to see. Okay, I think I have control now. So thanks again for the introduction. I'm very honored today to talk to you about our progress on gene editing approaches to PKU. And I want to absolutely thank the National PKU Alliance for their generous and continuous support of our efforts towards developing a cure for PKU. And I like to start with my acknowledgments, otherwise I forget to get to them. I don't do this work alone, and I have a lot of collaborators around the world that have contributed to this work. And today I'm going to specifically highlight the work of Dalen Richards, who was a graduate student in my lab whose work was entirely supported by NPKUA. She's already gone off to med school, but she's back in my lab for the summer, and she's defending her PhD thesis in August. Secondly, I'm going to present some very early preliminary data from Ann Boneta, who is a student in the laboratory of my longtime mentor and collaborator, Dr. Marcus Grompe. And she's been doing some experiments with in our mice using a novel lentivirus vector that was developed in the laboratory of Dr. Luigi Naldini in Italy. And again, we're very grateful for the continuous funding of the lab from both the National Institutes of Health and the NPKUA. So the standard approach to liver gene therapy for PKU for many years has been gene addition, in which a copy of the phenylalanine hydroxylase, or PAH gene, is simply added to liver cells without really altering the patient's own DNA. So the most common way that this is currently being done is you deliver the PAH gene using an adeno-associated or AAV virus. The one that we use mostly in the lab is what's called serotype 8. There are various different flavors of AAV, and 8 is the one that works best in mice. There's different ones for humans, as we'll see in a minute. And we and others, we did this more than 15 years ago now. And in this gene addition approach, this piece of viral DNA goes into the liver cell, doesn't really associate with the host cell's genetic material in any way. And once it's in there, it starts making its messenger RNA and ultimately leads to production of PAH protein and lowers the blood Phe. Now, based on results of lots of different laboratories, experiments in mice, there are now 2 different gene addition trials using AAV vectors that have been launched in adults with PKU. So Homology Medicines has dosed 4 adults to date. I don't have any insider knowledge about these trials. It's just data from a press release that they had from December. They've dosed 4 adults, and they've had some promising early results in a couple of patients. BioMarin has a different flavor of AAV that they're also planning a trial with. I think both of these trials have been slowed down somewhat, though, by the COVID trial, and really looking forward to seeing the results of these trials. They both have slight differences in their vectors, and it'll be very interested to see if there's any advantages to one over the other, and I don't have a strong opinion about either at this point. There is, however, an inherent limitation to this approach. As I mentioned before, the vector DNA that is introduced to the liver cell doesn't associate in any way with the host cell DNA. So if something causes the liver cell to divide, the cell's genetic material is copied and carried through to the daughter cells, but there's no mechanism to preserve the gene therapy vector DNA, and it gets lost. So the liver has the unique capacity of being able to rapidly heal itself after an injury or an insult. And normally, this is very beneficial for us, but it might not be so useful if you've had gene therapy. So liver cells in adults gradually turn over and they're replaced. And it's perhaps even faster if you're very fond of martinis or scotch. And in mice, if we do something to damage the liver after gene therapy, like if we remove part of the liver, the remaining liver immediately begins to regenerate and we lose the gene therapy right away. And we don't really know how long gene addition from AAV will last in adults, although there have been adults with hemophilia that have been treated with AAV that are out more than 10 years who've had stable function. But we really don't know how long this will last in humans, in adults. So furthermore, this approach won't work at all in kids because their liver's growing so fast that it will immediately kick it out. Now, no kids have been treated, but we know this from experiments with animals, including PKU mice. If we inject AAV into a newborn PKU pup, it lasts about a day and then it's gone. So how do we design a more permanent gene therapy? And that's really the current focus of our work. So what if we could just edit the patient's own DNA and change it back to the correct sequence? So instead of gene addition, what if we could find a way to go in and physically fix one of the patient's PAH genes and restore their own PAH production? And this is what we call gene correction. There are a few different methods available to try to surgically correct a gene. But one of the hottest technologies in the field is gene editing using CRISPR-Cas9. You might have already heard about this. CBS 60 Minutes did an entire episode on this. The technology was first applied to mammalian cells back in 2013. So this is very new, but there are thousands of papers already about this technology. But there's also been a lot of hype and I think a little bit of misunderstanding about it. And this is what we've done with it. So we have taken the same AAV8 virus and we've swapped out the PAH gene and put in the gene for Cas9. So Cas9 is a protein that bacteria have, and they use it to defend themselves from foreign invaders like viruses. Bacteria have viruses also that make them sick. And Cas9 is normally in bacteria, and Cas9 uses it, the bacteria uses Cas9 to locate foreign DNA and to cut it and kill it. And it's very discriminatory. It seeks out very specific DNA sequences and cuts the DNA. The beauty is that we can engineer the system to cut in very specific site close to the mutation in the PAH gene and cut only there. So Cas9 will frequently cut both chromosomes, but I'm only showing it just cutting one. So Cas9 just makes a cut and leaves a hole. And somehow a lot of people, even a lot of scientists, have somehow misunderstood that CRISPR editing somehow can cure a disease, but that really isn't true. It just makes a cut in the DNA and it doesn't fix anything. The cell itself has mechanisms to fix the hole, and we've learned that liver preferentially actually just sticks the two ends of the broken DNA back together to patch things up. That doesn't cure anything. In fact, sometimes it makes things worse because it just basically wipes out a big chunk of the PAH gene. This technique is actually useful for making new disease models, and you'll learn later from Erik how they used this to make a new PKU pig model. We've recently used it to make a brand new PKU mouse. Anyway, to get back on track here, how do we get a fix instead of a big mess? We infect the cell with a second AAV that carries a piece of the normal PAH gene without any mutation in it that matches the area of the cut. Now, we've left out any of the signals that would tell the liver to make PAH protein. And furthermore, it's not the whole PAH gene, it's just a little tiny fragment. We add this fragment, and our hope is that in the course of repairing the whole, the cell will incorporate this piece of the PAH gene. And if this is done correctly, the patient's chromosome now has an intact full-length PAH gene without any mutation that now encodes the correct sequence for the PAH protein. So Dalen's work on this was just published last month in the journal Molecular Therapy Methods in Clinical Development. And this figure here on the left shows the diagram of the genomes that she built for the two AAV vectors. This first one is the AAV that makes the Cas9 protein. The second one has the PAH repair template, and it's a piece of DNA that is analogous to the mouse DNA that has the PKU mutation, except it has the normal DNA sequence. It does not have the mutation. Otherwise, it's pretty much similar. The second figure shows what's supposed to happen. The Cas9 is supposed to come in here, and this is the DNA from the mouse liver. The Cas9 is supposed to come in here and make a cut. It doesn't cut right at the mutation. It cuts at a very specific site downstream. What normally happens is that the liver itself, the liver cells, have mechanisms to fix this cut. And the preferential thing that it does is to do what's called non-homologous end joining, or NHEJ. And it would just go in here and fix this cut down this pathway on the right. That doesn't really help us because you just get the same DNA sequence that you start with. And the alternate pathway is what we want. The alternate pathway is called homology-directed repair. In that sequence, then the liver would take our piece of DNA that we gave it and incorporate that and fix the mutation. And this HDR is what we're trying to get. So this is how the experiment is how she did the experiment. So she injected neonatal mice. So this is an important point. This only has worked in neonatal mice, as opposed to gene addition. This works in neonatal mice, and this has not worked in adult mice, probably because NHEJ works really efficiently in adult mice, and the homology repair has not worked in adults, and it's only homology repair has only worked very well in neonatal animals. So we've injected the vectors, the two vectors together in baby mice. She's redosed them here. She weans them, and then she does it again when they're basically teenagers. we probably don't need to do this, but that's what was done. And then later on, she euthanized them at various time points to analyze them, except in 2 animals, a male and a female. She let them survive, and 2 of them actually she let live out to 65 weeks, and she was able to actually show that pair was able to breed and give us pups, which normally we never see in PKU mice because of a maternal PKU effect. So she's actually able to overcome the maternal PKU effect by treating them at birth with this method. So that was very successful. Just making sure I cover everything. Now, when she first did this experiment, she injected the 2 viruses, and she found that no mice had been corrected at all. Nothing happened. And when she looked at their liver DNA, there'd been no corrections. And what we learned was that the Cas9 had cut, but then the cuts had been rapidly repaired by NHEJ, and we got the same sequence back. It looked like everything had been, the DNA had been completely uncut or untouched. And the trick that we learned, if I go back, is that we actually had to give them a drug, vanillin, which actually comes from vanilla. And vanillin was known to impair and block NHEJ. And then the cells don't have any choice. They have to use HDR. So the trick that she learned to do was at the same time that she gives some of the animals the virus, she gave some of the animals vanillin. And it was only then that she started to see significant amounts of HDR happening. So that's in this chart is when she looked at the DNA of the mice, the DNA in the liver. Here this is the what's happened to the DNA. In blue, she's looking for pieces of DNA missing from the PAH gene. So this is not corrected DNA. This is holes in the DNA. This doesn't help us. In black is where we've actually incorporated the normal PAH gene. So this is what we want. And this group is RTAAV plus VAN. These are animals that only got the repair template. They did not get CAS, so they didn't get cuts, but they did get vanilla. So this is sort of our control group. This group in the middle got both vectors, but did not get any vanillin. So they got CAS, they got lots of cuts, but they didn't get much correction, no HDR, because they didn't get any vanillin. It's only when we gave them vanillin, they got lots of cuts, they got lots of Cas9 cutting and holes in their DNA, but only when they got vanillin did we start to get some HDR repair. And what happened then on, I gotta make sure I get the numbers right here, is about 13% on average of genes got the correct repair, and that then translated in these animals to a mean of about 10% wild type enzyme activity in the livers. And that ended up giving them a range of Phe levels from about 250 in the best case scenario up to about 1200, but typically these animals run up here around 1500 to sometimes over 2000. So not a complete correction, but a big improvement. And our biggest limitation still, if you go back to looking over here, is we have a bunch of DNA that's been cut and damaged, but not still with incorporating our DNA repair template. So we still have a ways to go to try to get our repairs into these holes that CRISPR has made. That's our biggest thing that we're working on now to try to influence that ratio. And the PKU mouse, as many of you know probably, is tan colored because Phe inhibits melanin production, and the gene edited mice all turn black because we did lower their Phe levels enough to be able to get the coat color change in them. So that's at least satisfying. But so a very promising outcome, but not perfect correction just yet. And so we have a plan, next set of experiments planned to try to influence the ratio of cuts to repairs and try to improve that. That's our next goal. So switching to the next experiment, the one limitation of CRISPR is that you basically have to redesign your repair template for every different mutation that a patient could have. So there's over 1,000 different mutations known in PKU. So that basically would mean that we'd have 1,000 different drugs almost. And that's going to basically be impossible to do. Now, there are different ways of trying to approach this, but one of the ways that we're looking at is basically a combination of gene addition and editing, where we essentially build a genome that has the entire PAH. It's not the whole gene, it's called a cDNA, but it's a complete PAH sequence. And instead of having it just being added, we would have it integrated directly into the genome. And lentivirus can do this. And that's the experiment that we're now doing with the Gramignoli lab. And I don't have time to explain exactly how this is done, but the lentivirus does not go into very many cells and has very few integration events. We have to build in a system to allow us to select and expand that population. This is the experiment that Ann Veneta has done recently, but she has been able to get selection of enough cells that we've achieved about 5% enzyme activity with this method. And it has allowed mice to turn black. And in the 3 mice that she's done, she's got their blood Phe levels to drop down to almost the normal range, not quite. So this is a very early experiment with very preliminary data. It does not matter what the mutation would be in this case, that whole gene gets integrated someplace into the liver DNA. We've not done any molecular characterization to look to see where the gene is actually integrated, so we have a lot of work left to do. And again, this is a very early experiment. So this is our overall goal. It's to develop a safe, effective gene therapy method that will really yield permanent expression in a majority of liver cells, permanently reduce the blood phenylalanine, and hopefully lessen the dependence upon dietary Phe restriction. And hopefully that will give you some hope for the future of a cure for PKU. And I'll answer any questions at the end of the session. Thank you for listening.
Thank you, Cary. It's very, very interesting to see how we are able to, at the very level of DNA, actually make changes that can hopefully someday lead to a cure and improve the lives of PKU patients. Questions for Cary can be asked through the chat function, and we'll try to address those at the end. Next up is Dr. Erik Koppes at the University of Pittsburgh, who is going to be talking to us about edited swine and cell culture models of PKU. Erik, welcome to the webinar, and we're looking forward to learning about the work that you're doing.
Oh, well, thanks for the introduction, and it's great to be here to tell you guys some of my research that's been supported by the NPKUA during my postdoctoral fellowship. So I'm going to tell you about some genome-edited models, one in pigs and another that I've newly developed in a HepG2 hepatocyte hepatocarcinoma cell line. So it was great to have a kind of an introduction on CRISPR genome editing from Dr. Harding, and I'll try to be brief about my introduction on CRISPR since he gave such a great overview. Oops, may have clicked a little bit too many times. So just a little bit of background. I'm sure you guys all know that PKU is fundamentally a monogenetic disease. That is, it's caused by genetic mutations in phenylalanine hydroxylase. Of course, there's a few, there's a minority of patients that have mutations in the tetrahydrobiopterin recycling pathway, but the majority of patients have mutations in PAH. And so PAH, of course, is the enzyme that converts phenylalanine to tyrosine. And without that function, you get very high levels of Phe, upwards of 1,200 micromolar in classical PKU. And this is kind of termed hyperphenylalaninemia, although there's a range from moderate to severe. And so the ENU2 PKU mouse, which Dr. Harding talked a little bit about, is kind of the most widely used mouse model for PKU research. I mean, it's a great model. It biochemically captures many of the aspects of PKU in terms of from hyperphenolemic to lower tyrosine levels to some of the neurotransmitter defects. And of course, it's been used to rescue in early AAV preclinical trials. And so I've also shown the PAH structure. This is the rat structure solved by Arturo and Jaffe. So there's other models, including one in rat and in non-human primates that use kind of a Phe analog that inhibits PAH function. So these are really not genetic models, but they have their uses as well, because you can kind of study the effects of hyper-Phe. And we have, these rodent models are pretty good in the biochemical aspects, but they lack some of the major neurobehavioral deficits. There's some evidence for executive function deficits in the mice, and it's a little bit subjective, the behavior assays in mice. So so we're trying to build new models that may better recapitulate these complex neurobehavioral deficits. And so we thought a large animal model may better recapitulate these phenotypes of untreated patients, as well as early treated patients that become off diet. So a little bit of background, we chose the pigs. So pigs are a large animal model. Their size, physiology, and metabolism are closer to humans than mouse. Their brain structure and development is pretty similar to humans as well. And in the lower right, we see a panel from Conrad and Johnson, where they show that the pig and human brain development postnatally tracks order of magnitude off, but they track in terms of the timeline and progression of this kind of early postnatal growth of the brain. As well, you can do neuroimaging studies like what Dr. Shawn Christ has done about using DTI, so you can get mapping of white matter, also do gray matter function analysis. And of course, pigs are able to have a very complex neurocognitive abilities. We've all seen the movie Babe, where they train a pig to be a sheepdog. And the genomics and gene editing has really come into its own for pigs in the past decade. So the pig genome sequence is available, and the genetic tools to make mutations to create different models has really made it possible to make PKU pig. So a little bit about CRISPR-Cas9, as Dr. Harding mentioned, it's adapted from a bacterial system used to cleave integrated phage genomes. But basically, you can think of it as a molecular scissor. So it cuts DNA. And how it does this, it does it in a sequence-specific manner. So Cas9 is the nuclease enzyme that cuts the DNA, but it's guided by these short RNA species that are complementary to genomic regions. So the complementary region of about 20 base pairs is used to target to specific locations in the genome. And that brings the Cas9, and it makes a double-stranded DNA break. And so these DNA breaks are then repaired either through non-homologous end joining, leading to insertion deletion mutation indels, or if you have a template available, you can get HDR, in which that template is integrated in. And you can also make two different cuts and delete out regions that are intervening. So if you have two adjacent cuts, say 2 kilobases apart, 2,000 base pairs, you can get larger deletions rather than these small indels. And so that's what we chose to do. And in looking at the PKU variants here from BioPKU, you see that exon 6 is really the exon with the largest number of variants. And in particular, if there are some patients with exon 6 completely deleted, and that results in exon 5 to exon 7 splicing, and that results in a nonsense-mediated decay via early translational termination. So basically the protein's shorted out and it doesn't have any functional activity. So we chose to design guide RNAs within intron 5 and intron 6. If we get to the next page. So these guide RNAs 5.1 and 6.2 are within intron 5 and intron 6 on either side of exon 6. And so we did zygotic genome editing. This was with the National Swine Research and Resource Center in Missouri. And they injected guide RNAs that we had previously screened in a cell culture model, along with the Cas9 messenger RNA. So messenger RNA became protein, the Cas9 protein, and formed a ribonucleic protein complex with the CRISPR guide RNAs within the pronuclei, and these then were recruited to the PAH locus and performed gene editing. And so we cultured those one cell embryos to the blastocyst stage, and then screened them and found that many of them had deletions. So in that panel B, you can see that many of the lanes have a PCR band indicating a deletion. And these were then transferred to a surrogate sow, and we ended up generating a couple of genome-edited piglets. And so we actually have 2 genome-edited PKU pigs. One is a homozygous affected animal, and one is a heterozygous carrier. So in A, I'm showing kind of the schematic of the genome editing. So the pink boxes flanking exon 6 are the guide RNA sites where we made the cuts. The yellow boxes indicate these PCR amplicons used for droplet digital PCR that were used to assess copy number. And A, B, and C are these primers used for genotyping. And what we found in B was that the copy number for exon 6 in our homozygous animal 1161, the red bar graph, goes to 0 in exon 6, whereas in the heterozygous animal 1162 in blue, that drops to a copy number of 1, indicating a heterozygous deletion. And then for exon 7, curiously, we found that 1161 also had a heterozygous loss. So it indicated a compound heterozygosity, with one deletion having just exon 6 deleted and the other having exon 6 and 7. And this was confirmed by PCR, shown in part C. We also did a whole genome sequencing to 30x coverage, and that's shown in panel D. And so basically those blue bars are kind of the terrain along the PH gene, show the sequencing reads mapped to that location. And so the Yucatan's a control, and so you can see that the peaks are, while they vary a little bit, they're pretty consistent across the gene, indicating it's completely intact. Whereas in 1161, you can see the complete absence of reads within that exon 6 deletion region, and extended a heterozygous region with kind of a low terrain profile through intron 7, indicating that heterozygous compound heterozygosity. Whereas in 1162, you get the heterozygosity just between the 2 guide RNA sites, indicated by the red triangles. So onto the phenotypes for the PKU pigs. So the most obvious phenotype from their birth was that the affected animal 1161 was growth restricted neonatally and postnatally through the first year of life. So you can see that in figure A, as well as the chart in figure B, 1161 being the purple graph. So she did eventually catch up at around 1 year of age and is about the same size as 1162 currently. But the growth restriction is certainly a phenotype for untreated PKU. We also did a dietary Phe treatment for the affected animal. So it should be noted that her Phe levels are extremely high. So it's like 5,000 micromolar pretty consistently, 4,500 to 5,000. So this is really high compared to the mouse, where it's 2,200 to 2,500, depending on the sex of the mouse. And in humans, where in classical PAH deficiency, the levels are around 1,200 micromolar or greater, but rarely that much higher. So our affected animal during a Phe washout. So we had a specialty diet where there's absolutely no Phe in the food. For 2 weeks, her levels came down from 5,000 all the way down to 100. And that's in graph C here. And then we switched the diet after seeing the efficacy of the washout to 50-50, and she kind of spiked back up, and we changed it to a 75-50, and then eventually to an 85% Phe-free chow. And her Phe levels, again, blood Phe levels gradually came down to therapeutic range. And we repeated this washout again, just doing the straight 2-week washout, and then right to the 85% Phe diet, and that consistently had had her in the therapeutic range. And you can see a little blip at the end, and that's just may have been due to a measurement that came right after her feeding in the morning. What's also interesting is that we did some MRI imaging of the affected animal and a control pig. So the control is A and C, and the affected animal is B and D. So she actually had a smaller brain overall, but also notably, and it's not completely clear from these images, but you'll have to take our word from our analysis, is that we had a reduction in cortical gray matter in the PKU affected pig in terms of the normalized brain volume. So this is a pretty intriguing finding, and I think Shawn is pretty intrigued by it and has helped us out a little bit on interpreting the data. And so kind of, right. And we also had a litter of maternal PKU swine. So we bred the affected animal off diet and had a litter of 6 piglets. We also had, there was one stillborn and one spontaneous abortion. So there's kind of some analysis ongoing right now, post-mortem, looking at any neurological findings for neuropathology. We didn't see any cardiac defects per se yet, and we don't have a control litter, so it's not a perfect experiment, but it is kind of a pilot, and kind of the idea of what we could do if we had more PKU pigs to work with. And so that kind of leads us to our current and future directions. Right now we're breeding the affected and the carrier pig to a Yucatan mini pig. So that's Pumbaa there, that we named our guy. And the idea is to generate cohorts of PKU affected and control animals by first generating an F2 that would be heterozygous and intercrossing them. And we would like to continue to do a maternal PKU study on a mini pig background, and also to have full cohorts to have to do real statistical comparison for neurobehavioral and neurocognitive studies and investigate alternative therapies. And briefly, I want to tell you guys about a HepG2 cell model I've been working on the past year. So HepG2 is a commonly used liver cell line. It's immortalized, it grows very well in cell culture, and it expresses high levels of PAH. So it's the perfect genetic model system to use for PKU research. So I designed CRISPR guide RNAs to knock out the whole gene. It's about 75 kb, so it's a pretty big deletion. So I designed them guide RNAs intron 1, intron 12, transfected them into the cell line, and generated a few deletion lines. So clone D3 came up in my initial screen, and actually has a deletion and one allele and an inversion on another, but the effect is that it has no PAH protein. And then I got 2 heterozygous null lines, which I then transfected again with alternative guide RNA pairs, and got 2 additional null lines, one has 2 nulls, and another that has a null and inversion. But the effect is the same, that they have no PAH protein. You can see by the immunostaining the lack of green in panel E for the deletion line. And my plans for the HepG2 model include doing gene expression analysis through RNA-seq and control for PKU cells to see if there's a gene expression changes based on the genetic difference, and also to do that in complete media versus media that's spiked with 1200 micromolar Phe to see what the effects of hyperphenylanemia would be on these cells. Also doing some mitochondrial respiration, oxidative stress, as there's some studies indicating that these pathways might be involved in mediating the phenotypes of PKU. These models would also be really good to knock in patient-specific pH variants, kind of like what Cary was talking about, but instead of rescuing with a wild type or a normal pH variant, you'd knock in one with a patient-specific mutation and be able to screen different compounds or develop different reagents to treat. The model could also be used to develop gene therapy reagents. So that's kind of my future directions, and I hope to tell you guys more about that in the near term. With that, I'd like to just acknowledge the NPKUA, and in particular the Stevie family, who's supported me for 2 years through their generous family foundation, and my PI, Robert Nichols, who's also has been supported by the NPKUA to develop the pigs. Marie Johnson, who's a great lab tech, Jerry Vockley, who's been a great co-mentor, Steve Dobrowski, who's really kind of tutored me on the ins and outs of PKU, and of course the National Swine Resource Center for developing the pigs with us. And just wanted to give a quick plug for a study that Jerry is doing here at Children's Hospital of Pittsburgh. He's doing a hepatocyte transplant, so if you're interested, you could talk to Jessica Lindenberger. Her email's there at the bottom. I'd just like to thank you for your time, and if you have any questions, please feel free to ask, and I'll answer them at the end.
Lex, you're muted.
Thank you, Desiree. Just wanted to say thank you, Erik. I had some other things to say. I'll rack them up at the end. But in the interest of time, I think we're going to move to Desiree rather quickly. Welcome, Desiree.
Thank you. Let me gain control of my screen. I believe I have it now. So what I'd like to talk with you about today is advancing clinical drug trial endpoints. And what this means is how do we get to some better ways of determining whether a drug is working or not for people with PKU. This work actually sort of had its genesis in a meeting that was a conference that was put on by National PKU Alliance, thank you very much, where we talked about sort of where we were in terms of the tools that we use in clinical drug trials for PKU. And one of the things that came out of that was that we know there have been a lot of advances made in the treatment of PKU in recent years, particularly within the last 15 years or so, right? But as our own Christine Brown found through surveying people with PKU, talking to a lot of folks, most people with PKU believe we still need new treatments, or we need to tweak, improve the treatments that already exist. Now, one thing to keep in mind is that when we try to get a new drug approved for the treatment of PKU, it's a very lengthy process, actually. And this is a process called clinical drug trials. And I'm sure that many of you have participated in some of these clinical drug trials. And during the process of those trials, we need to have some sort of endpoint. So in other words, what's telling us whether the drug is working or not? Or it may not be a drug. I'm using that as sort of a generic term. It could be liver cell transplant, hepatocyte transplant, or something along those lines. But whatever the treatment is it working or not? So what is the data point that tells us whether the treatment is working? And up to this point in time, primarily that data point has been reflected in Phe levels. So, you know, Phe levels are high in people with PKU. Does the treatment bring down Phe levels? And that has been our primary way of deciding whether a drug is going to help people with PKU or not. Now, the Food and Drug Administration, the FDA, has suggested that that's great. We certainly want Phe levels to come down because Phe levels are correlated with all kinds of things, including cognition, findings that we see on brain scans, growth, all kinds of things related to our phenylalanine levels. But the FDA has also suggested that as we move forward, our endpoints, or how we determine how much the drug or the treatment is helping people with PKU, needs to reflect something that is also functional. So not just are Phe levels coming down, but are the lives of people with PKU actually improved in relation to those reductions in Phe levels. So in our conversations at the NPKU conference around these issues, we talked about how cognition, or your thinking abilities, is an area of great functional concern in individuals with PKU. Now, I'll say right off the bat, there is a great variability here. Some people with PKU, their cognitive abilities, their thinking abilities are within the range that one would expect. For other people, they have more problems, though, in these areas. But cognition, or thinking, is obviously quite critical to our day-to-day function. So this would be, in functional terms, a good endpoint to look at in clinical drug trials. If we want to know if it's possible that the lives of people, the day-to-day functioning of people with PKU will be improved by a treatment. In thinking about cognition in more detail, we settled on a few areas of cognition that we wanted to examine for this purpose in particular. One is IQ, or general intelligence. That's just a broad, overarching term that tells us what somebody's intelligence level is. More interesting, actually, are looking at specific abilities that go into making that general intelligence. One of those is executive abilities, and that's been alluded to in a couple of the other talks. That's higher order processing. How well can you plan out what you're going to do? How well can you hang on to information as you process that information and do something with it? There are other things like inhibitory control or impulse control that go into executive abilities too. So it's sort of this broad category that captures a lot of things that help us organize our day-to-day function in the world. And then another area of great interest is processing speed. So that's how quickly can you do stuff? How quickly can you think about stuff? So executive abilities and processing speed in particular are areas that people with PKU have more difficulty with on average. So these were the two areas of cognition that we settled on for good reasons in terms of thinking about phenylketonuria and whether people are being helped at a functional level in clinical drug trials. Now, I'm going to give you an example here and let you do a little work for a few minutes. Maybe this will serve as that cup of coffee that you might need at this point, having been very attentive for quite some time now. This is an example of one little component that one little test that we might use for a test of executive abilities. And this is called a matrix reasoning task. You'd actually look at these puzzles and you might do 25 of them. So it wouldn't just be one either. Some of you may have been in some of our studies in the past, or other folks' studies where you've done cognitive tasks similar to this. So the idea is that you look at the 4 images in the top here, and one of the boxes, one of the images is simply a question mark, and your task is to look down below and determine which of these fits into this box, given the information you have in the other 3. So I'm going to give you just a minute to look at that. but not too long or we'll all get really bored. Maybe you figured it out, maybe you haven't. This is sort of an intermediate one that we might present to someone who's in one of our studies. They get so hard that it takes minutes and minutes for people to figure out how to do them. So if you can reason through how to do this, we can get a score of how many you can do correctly, how many are incorrect, and from that determine sort of on average what your executive abilities look like. There are a number of other tasks. This is not the only way we assess executive abilities. We use a number of tasks to come up with sort of a composite or a combined look across impulsivity, across this sort of organization and planning, strategies, holding on information online while you compute a task, for example. We use all of that and put scores together to get a big score that tells us in general what someone's executive abilities are like. And I'm not going to give you the answer to this. We can take a poll and see how many got it at the end during question and answer. Okay. Now, there are a number of challenges in terms of using cognition as a clinical trials endpoint. At present, there is actually no cognitive tool, no instrument, no particular test that has been validated for the specific purpose of use in PKU clinical trials. We need such a thing. We badly need such a thing. This validation along these lines just means that we determine it's a really good thing to use for this purpose. We need this because there are a variety of studies that are going on, that will be going on in the future. And if we have a tool that could be used across studies, it's going to be a lot easier for us to think about how the findings from one study compare to the next study. It's going to be a lot easier for investigators to simply know a tool that is good, that has been validated for this purpose, and go to that instead of trying to reinvent the wheel every time we have a clinical drug trial. Excuse me, my hay fever is getting to me a little bit here. So this is an important task that we're undertaking here. Now, the problem with the existing cognitive tools, any of you who have been in some cognitive studies probably know that the test can take a long time to complete, like 3 or 4 hours, sort of, if you're lucky. And that's a pretty big burden. We have a very dedicated bunch of folks with PKU who want to contribute to studies and do contribute enormously to studies to better the lives of people with PKU. But let's face it, we all have lives. We have other things to do. We have kids to raise. We have jobs to go to. We have school to go to. It takes a long time to be in these studies. So I just want to thank everybody who's been in a study and tolerated this. So we wanted to try to find a tool that takes less time. A lot of these tools are really expensive to use also. One of the tools we're using in our study, and I'll get to why we're using this when I'm griping about it being expensive, costs thousands of dollars. We want to come up with something that costs hundreds of dollars so more people can use it across more institutions, and it will just be more easily available to folks. A lot of our cognitive tasks and tests require specialized training, or you need some sort of specialized equipment to do it. It's also possible that there's not normative data. That means data collected on a particular test from a really large sample of people across the United States, across the world, that gives you an idea of what, on average, an average person without PKU, how they would score. And so that is a good comparator for us. How do people with PKU compare in terms of their scores with people who don't have PKU? A lot of the studies that have been done around cognition and psychological problems as well relies on an individual saying, I have this problem or I don't, or let's say a mom or dad saying, I do have the problem or don't. So that is not always the most reliable. We like to use a mixed method for looking at that information. And then, you know, a lot of tools aren't standardized. They aren't automatic. They're just problems and rolling them out for multi-institutional research. So we have selected the NIH Toolbox as the measure that we would like to validate for purposes of clinical drug trials in PKU. And this assesses the key areas of cognition I was talking about: IQ, executive abilities, speed. It takes only 35 minutes to complete, which is fabulous compared to 3 or 4 hours. It identifies change over time, has a big normative sample. It's easy to roll this out across a lot of institutions who might be involved in a clinical drug trial. PKU is a rare disorder, so very often we need to come together as consortia, come together as multiple institutions in one study to do the best research possible. So we have settled on the NIH Toolbox. It's also very widely available. The National Institutes of Health put this together, and it costs about $500 a year. So this is much, much less expensive and accessible. It's online. It's done on an iPad for the most part. So it's a really great tool that we're trying to validate here for these clinical drug trials. So our hope is to have 150 individuals between 8 and 55 years of age who were early treated for PKU. And we're going to look at how people perform on the Toolbox, this new, fairly new instrument, compared to how people perform on a lot of other tests that have been in PKU research. So if you're in this study, it's still going to be the 3 or 4 hours because we have to determine how well the Toolbox does in comparison to other tests that have been used in PKU. But we hope that in the future then this will greatly reduce the time that it takes people to do the cognitive tasks in clinical drug trials. I've listed some of the tasks here. I'm not going to go through this, but I put it here for the sake of thoroughness in case someone would like to take a look at the specific tests we're looking at. I'm also happy to offline discuss that with anyone. So that's what we have going on here. I would again like to thank the National PKU Alliance for the support in doing this project. We're moving along. We have had a slowdown due to COVID. I haven't been able to be in my lab for months, literally, but we're all working remotely from home and bringing this study along. The last thing I'll say is if you're curious about the little pictures I inserted here of the brain, these are from kids who sent me pictures they had drawn as parts of their thank you notes after I had taken a brain out to their school. And I just, they make me happy, and I hope they've made you happy to take a look at them today too. That's what those are. So I'll send it back to Lex, and thank you very much for your attention, and thank you for anyone who's been in our studies, will be in the future, and for your attention.
Okay, thank you, Desiree. So I think, you know, from this series of presentations today, you can get a pretty good overview of the diversity of approaches that are being taken out there to think about ways to improve the lives of PKU patients and those around them that love them. And I think particularly from Desiree, but I think this is highlighted through all of them, is that while blood Phe levels have been the most important and useful tool in the past, we have now, through the research of many, begun to understand that there are other things that we should be looking at in terms of measuring the overall improvement in the lives of PKU patients. We realize we're running over a little bit here, or about to run over, let's say, and that during the time that folks have been presenting, there have been questions that have been flowing in, and I'm going to try to feed those questions out, if you will, to the folks. So maybe the first question that came in was from Natasha, and she was actually directing to you, Shawn. And Natasha has noted that in the research, or there are many similarities between the research of the brain structure, if you will, white and gray matter, between PKU patients and those with other forms of brain disease, such as dementia and Alzheimer's, for example. Is there good communication between those two disciplines to look for synergy, if you will?
Lex, Natasha, it's a great question, a great topic and point. It's something that in our lab we've started to explore as we're working with I don't call them older adults with PKU, but older than the children that we often work with. So, our adult study, we started to look at some commonalities, potential commonalities. We know that one of the brain regions that's particularly affected in Alzheimer's is the hippocampus, which is very important for encoding new memories. And so we've talked about, and within the context of our current study, starting to look at that brain region in particular to see if there are really commonalities. Another thing that we're doing is kind of relates to the fact that with Alzheimer's, you can often see brain atrophy. And the way that shows up in the MRI is often the space between the skull and the brain basically is bigger, right? That's with a smaller brain because of the atrophy. And so we're looking at exploring that as well in this new data set. But again, great question. It leads into a bigger question of, exploring the ideas of what does it mean to grow older with PKU. So we are definitely looking at that, and it was a great question.
Thank you, Shawn. Roberto, a question that came in for you was based on your experiences right now, do you think that these AE cell treatments can we get enough cells in there to help patients? Do we have any insights into the durability of that treatment? And of course, the extension of durability is, do we believe a patient could receive ultimately maybe multiple treatments as necessary? So kind of a three-part question.
Yeah, thanks for the question. Actually, it's a very good one. So it's required, I try to make it short, but require a very detailed part. So in all the patient we transplanted with liver cell, we know we need to do multiple injections because we need a large amount of cells. Liver is a quite large organ, so with billions and billions of cells, so we need to infuse a lot of cells. Once said that, we know that for the hepatocyte transplant, it was requiring several amounts because we could not preserve cryogenically. We cannot freeze the cell, having those available all the time. With the placenta stem cell, is actually quite simpler. It's a little bit easier because we can freeze the cell, having them ready to transplant the patient. So we can infuse different times or multiple time infusion, but it might take few days or a few weeks potentially. How long this cell lasts in the human body, we don't know. There have been done 6 patients so far, but nobody has ever dared to test the patient to see the cell still there. So usually is an indirect measurement because we measure the indirectly if the disease is corrected is due to the cell. The same we were proposing to do with phenylalanine level, for example. We measure the phenylalanine level in the serum and monitoring how long the cell can last. We know from animals that the cell lasts very long, these cells, because we find out that the animal, at the end of his life, we use the animal, we sacrifice that, and we found the human cell still in the animal. So there are some indirect evidence that the cell might last very long. How long, we cannot prove it yet. So we are looking forward to the clinical trial to have potentially an answer for that.
Great, thank you, Roberto. Cary, this is kind of a horse race question for you. That you know, those reading the lay press, the non-scientific journals, you know, are aware that, you know, or maybe perhaps view gene editing and gene therapy as kind of competing technologies, and you know, I wonder what your thoughts are on that.
I hope that we'll have multiple different options available at some point. So, I mean, ideally, I mean, gene editing would be more permanent. The issue we have with gene editing is still getting enough cells. So we have a long ways for this to work in liver. There are a couple of gene editing protocols in humans that have happened. So sickle cell anemia and beta thalassemia, there are clinical trials being done, but in that situation, the editing is happening in cells that have actually been taken from the bone marrow, and the editing's being done outside of the body and then been put back in. So it's sort of like having a bone marrow transplant, and in that situation, the cells actually grow and expand. We don't have that luxury really in liver, so we're a ways from being able to, you know, get a similar outcome. So, you know, we're a long ways from being able to get equivalent effectiveness compared to what's able to happen with gene addition. I'm not even going to put a timeline on it because that's a big trap. And the other issue I didn't even talk about that everybody's worried about with CRISPR is, if, you know, it's designed to cut where we want it to cut, but does it cut other places that we don't want it to cut? Right. We actually didn't find it in our mice. We didn't find any cuts anyplace else. Or if it did cut, it fixed them without any problem. But this is a big concern for any of the editing techniques, is what's called off-target cutting. There has been a lot of concerns about this. They see it a lot when they edit cells in Petri dishes. They see a ton of cuts other places, but I just think that cells in Petri dishes don't do as good a job of fixing things. In the animal, it seems to work. It seems to we fight the repair mechanisms, as you can see. It fixes it all back to the way it was and doesn't do what we want it to do, and that's what we're battling.
Great, thank you, Cary. Erik, a question for you. We all know that there are you know, just literally hundreds of different types of animals that can be used for clinical trials, and of course, people are all familiar with you know with mice and rats and bunnies. And I guess a question that occurs to people is that if you were going to try to pick an animal that would closely emulate human anatomy and physiology, a pig is not such an obvious choice to many people. Can you comment on that a little bit?
Sure. So in terms of the phylogenetic tree, actually mice might be a little bit closer in terms of evolutionarily being closer to primates and humans in particular. But pigs do have an advantage in terms of their genomic structure are largely more similar to humans, and you know their body and physiology, so their size is just and their metabolism is a little bit slower than mice. You know, mice are have very high fast metabolism, so pigs are a little bit slower, and they've been used to model other metabolic diseases. So it's good in that sense, and as well, mice are lissencephalic, so they have a kind of a smoother cerebral cortex, whereas pigs, as well as primates like humans, have a very convoluted gyrencephalic cerebral cortex. So their development is more similar, and there's a lot of postnatal maturation of the brain. So the timing of the brain development is actually a lot more similar. I would say, with a caveat, probably a non-human primate would be the best model for PKU. There's not as much work, or it's still a very new field, I think, for editing primates. I mean, there's only a few disease models right now where primates are available in, and I don't think that's actually even in the US so much. And we've only seen, been working with the pigs, I wasn't sure exactly what to expect in terms of a neurobehavioral phenotype, and I didn't see anything particularly obvious in terms of a neurological deficit or behavior compared to a little mate, but we really need cohorts of the animals to compare, like 3 and 3 at least, minimally, to do any statistics. So that's really kind of important. And just to add to what Cary was saying about the kind of off-target editing, so we did a whole genome sequencing of the pigs, and we saw no off-targets. And that's looking at, so there's predictions of where those off-targets could be by homology to the guide RNA sequences, and we looked at all of them by whole genome sequencing, and none of them were mutated, although we found a few SNPs. But editing in embryonic zygotes in humans, there's a recent paper that showed that there can be large deletions of whole chromosome segments. So that is definitely something to take into account. But if you're doing that in hepatocytes or something, that might be the minority of cells. Of course, you don't want oncogenesis or cancer to occur. But I think the off targets are rare and can be ameliorated by design of the Cas9 and CRISPR system.
Thank you, Erik. Desirée, I see this question as more of a regulatory question, but since it's kind of popped up in your discussion, I'm gonna pass it to you first. That the question was, how does the FDA communicate their recommendations about other endpoints to trials apart from Phe?
Yeah, that's a good question. And, as far as I know, it's not as straightforward as them sending out some kind of memo to everyone saying we need we need a functional endpoint to look at as well. But I believe, feedback from the FDA as different researchers go through the process of attempting to get approval from the FDA for a drug or a treatment to be used in PKU. The feedback is Phe is great. We need to know that, obviously, but we really want to know about these functional outcomes too. It's just feedback that different people, different researchers have gotten, different studies, trials have gotten over time from the FDA, that something more functional is needed as well.
Thank you, Desirée. Shawn, a question for you was that for the patients that are the PKU patients that are in your study right now, can we talk a little bit about how well controlled their Phe levels were? And for example, were your patients largely just controlled by diet? Were perhaps any of them on any kind of therapeutics? I'm expanding the question, by the way. Did you monitor Phe levels, or at least take a Phe level at the time that you did your brain scans?
Well, so it's a great question from Hans. All of our participants in our study had early treated PKU, so they're treated from birth. From that point, there was a big variance in Phe levels across their lifespan, including most recent. So if we look at most recent, and I actually saw that question earlier and pulled the data, they ranged all the way from 200 to 200. So there was a wide range of level of control. For us, that's a good thing. Actually, we were encouraging that because we're really interested in what's that relationship between different levels of Phe and the outcome in the brain. And of course, if everybody had, let's say everybody had really low levels or everybody had really high levels in our study, we could see PKU versus non-PKU differences. but it'd be harder to tease out really the role of Phe in that relationship. So we, in some ways, we targeted a wide range, and we got it. There are a handful of people in there that are on Kuvan. So of course, that's taken into account in terms of looking at their Phe levels since they were on Kuvan. None of the individuals were on Palynziq or other medications to help control the levels. I'm trying to think if there was anything else in this question, but again, it's a great question, and hopefully it makes sense as to why we actually were targeting a wide range.
Great, thank you, Shawn. Let's see. I guess a question. This might be a question, a combo question for Shawn and Desirée. In terms of physical changes in the brain, I wonder if there are any changes that can be made in real time, and this kind of goes to the treatment question too. That if you had a high blood Phe level today, and then maybe later in the week your blood Phe level was lower, could you see that in could you actually see that in structural changes? And before you answer, I'm going to translate that question for Desirée and ask the question that if a patient had a high blood Phe level in one day and they took some of the cognitive panel tests and then literally came back a few days later and had a lower blood Phe level, would we expect to see changes in that sort of short window of a couple of days? Shawn, I'll let you go first, and then Desirée.
I was going to defer to Desirée. No, it's a question. No. I mean, there have been some studies done with Phe loading, and again, I'll kind of defer to Desirée a little bit.
Okay.
Is that my reading of that literature is that actually, yes, you can see when you do the Phe loading, you can see changes, especially in those kind of general things like attentional focusing, and sort of call it mental fogginess going.
Mm-hmm.
So, from the brain side, what I would say is that we're probably not going to see structural changes in the brain related to that on a short term, but we'll definitely see functional changes. So how the brain's activation is, how the brain, different parts of the brain are working together. Because of course that's what feeds the outcome of the brain functioning is the behavior. So again, I'll defer to my colleague from the east side of the state.
We have a lot going on in Missouri.
Yes.
So just to add on a bit to what Shawn was saying, there's a study, for example, around attention, which I sort of lump in with executive abilities, that shows these shorter term changes that map onto Phe levels. And we hear patients talk about this all the time. The Phe levels go up. People can feel the Phe levels, that the Phe levels have gone up. If they've gone off diet, they went to that party, whatever it is, they can feel the result, right? So we know that that is a thing for sure, and that there can be these very short-term changes. In moving forward in studies, the technologies that we have now are pretty fascinating in terms of being able to get these momentary assessments of what's going on with someone. So you can now send questions out on someone's telephone, and they could answer 4 times a day, how do you feel your thinking is, right? Something along those lines. We do something a little more sophisticated than that, but along those lines. So there are ways that we can track across days now, rather than thinking we have an assessment this year and we're going to get another assessment next year. There are fabulous ways that we can start to track these things. There's still issues to work out, but that technology and those sorts of tasks are really coming along, and I think that is going to be a wonderful new wave in thinking about cognition in people with PKU. The other thing that I would say is you would think if there's a change in your thinking abilities from day to day, there'd have to be some kind of change in the brain, right? Because that's what's driving thinking. There's a lot more going on in the brain than we can see on a brain scan. There's neurochemistry is happening there, and that will result in these structural changes that we see in the brain, right? So there are these finer level issues that we can't right now track day to day that are driving that.
Great, thanks both of you. So we're out of time. There are more questions that we're not going to have time to address today. I'm going to take a stab at answering those and posting some of those answers on the website. We're going to post this webinar on the NPKUA website, and I certainly want to thank all the presenters today for taking the time to discuss their work. And one of the things that was interesting to me, or interesting is not the right word, but I was very happy to see was the amount of collaboration that goes on between this group right here in terms of referencing each other's work and talking to each other. I know the connections are much deeper than that into the PKU community, and that could really show up at the NPKUA convention, which was unfortunately canceled this year, where we'd have a much larger number of people, both funded and by the NPKUA, and also people who are not funded by the NPKUA who are doing important research out there. There was one question I wanted to just take a quick stab at, and that was relating to how do we find out about clinical trials. And I just wanted to point out to all the attendees that there's a very valuable resource called clinicaltrials.gov that's on the internet. It is the FDA's website that lists all active clinical trials going on in the United States today. And you can reference that site to find out what clinical trials are going on. I'll give a little sneak preview that the NPKUA is in the process of building a clinical trial finder tool that we hope to have on our website in the next few months, which will enable or facilitate PKU patients to find clinical trials that are happening and enrolling and be able to reach out to them. So with that, again, thank you to the audience for your attention and participation. Thank you to the presenters and for presenting the exciting work that you're doing. And we'll look forward to getting together again in the not too distant future.